নমস্কার। আমি ডক্টর অংশুমান দিকপতি। বিশেষ বন্ধু অমিত জানার অনুরোধে আজকে এই যে অনেক পরপর অনেক সপ্তাহ ধরে যে কোভিড নিয়ে আ আমাদের বিভিন্ন ডাক্তার বন্ধুরা উম আলোচনা করছেন তার একটা অংশ হিসেবে আজকের আলোচনা। অ্যা ধন্যবাদ অমিত এবং বাকি ডাক্তার বন্ধুদের আমাকে এই সুযোগ দেয়ার জন্যে। অমিত কে আমি জিজ্ঞাসা করেছিলাম যে আমি বাংলায় বলবো না ইংরাজিতে বলবো? তো অমিত আমাকে বলল মিলিয়ে মিশিয়ে বলতে। তো আমার ওটাতে একটু অসুবিধে আছে। তো আমি বললাম একটা ভাষা আমাকে বলতে। কোনটা বললে সুবিধে হবে? তো অমিত বলল যে তাহলে ইংরেজিতে বলাই ভালো। তাই সেটাই আ ইংরেজিতে বলাই ঠিক হয়েছে। So a very good evening in India, good afternoon here. We are going to talk about genetic susceptibility and COVID-19 today. So in previous discussions, my colleagues, Dr. Friends, they talked about different aspects of COVID-19. So today we will see if there is any relation or connection between genetics and the spread and severity of COVID-19. So since the start of COVID-19 pandemic several months ago, scientists have been puzzling over the different ways the disease manifests itself. They range from cases with no symptoms at all to severe ones that involve acute respiratory distress syndrome or ARDS, which can be fatal. So, question is, what accounts for this variability? Might this be because of our genes? So, coronaviruses have raised such questions for more than 15 years now. So, we know this SARS-CoV-2 or the novel coronavirus, which is causing COVID-19, this is not the first coronavirus, it's a family of virus. So, in researching the 2003 outbreak of SARS in University of North Carolina at Chapel Hill, Ralph Baric and his colleagues, they identified a gene that, when silenced by a mutation, makes mice highly susceptible to SARS-CoV. So SARS-CoV is a coronavirus that causes the SARS. So this is called TCAM2, TICAM2. The gene codes for a protein that helps activate a family of receptors. These receptors are called toll-like receptors, TLR, that are involved in innate immunity, the first line of defense against pathogens. Now our attention has shifted to SARS-CoV-2, the new coronavirus that is causing this COVID-19. And these TLRs, toll-like receptors, they have once again drawn researchers' interest. This time, to help explain the excess number of men who suffer from severe infections. Yes, right. So, in late April, in France, the COVID-19 intensive care admission, 73% of that was made up by men. Now, yes, Some of that can be explained by behavioral and hormonal differences. So men are generally outgoing, going to pubs, buddy things, hugging people. Yes, but genes may also factor into the mix. Unlike men, women have two X chromosomes. So that actually carry double the copies of the gene TLR7. Now we are coming back to that TLR, toll-like receptors. And that is a key detector of viral activity that helps boost immunity. Now, there are a lot of things we are hearing about blood groups. So it is said that, okay, some blood groups are more into getting COVID-19. And to be precise, some studies have found that blood group A is more associated with higher risk of contracting the virus, whereas type O offers the most protection for reasons yet to be determined. Now, this came out of a study in China. There was a study by Peng Wang and his colleagues in Southern University of Science and Technology in China. Of course, this is not peer-reviewed. Now, what they did was they compared the distribution of blood types among 2,173 COVID-19 patients in 3 hospitals in Chinese cities of Wuhan and Shenzhen. And they found this. Now, in ABO blood group system, type O blood is the richest in antibodies. So it has both anti-A and anti-B, which points to the assumption that maybe these antibodies are protecting against COVID-19. Now, in 2008, there was a study there was a model study in University of Nantes in France. So, the researchers showed that the binding of the virus protein S to a cell's ACE2, angiotensin converting enzyme to receptor, which is necessary for infection to take place, is inhibited by anti-A antibody. Though this study did not say anything about anti-B antibody. So this ACE2, by now, all of us, we know that this ACE2 is used as a doorknob by the SARS-CoV-2 or the COVID-19 virus to enter the cell. Now, a close relative of this S2 in blood pressure control is S1, angiotensin converting enzyme 1. So there is a gene called S1D gene, now, which is one of several genetic variants of the enzyme and is associated with low levels of expression of S2 gene. So I'll try to make it clearer. So this S1D gene actually reduces the S2. So now S2, ACE2 is used by the SARS-CoV-2 or COVID-19 virus to enter the cell. So what happens if ACE1D gene is more in the body? That means there would be less number of ACE2. So in turn, that will make it more difficult for the virus to enter the cell. So now this ACE1D gene, that actually differs from one country to another, particularly in Europe. So that raises the question whether the geographical distribution of this variant correlates with COVID-19 prevalence. Might it reflect the epidemiology of the disease on a global scale? So there was a study in Belgium, Ghent University. So they used data from 25 countries. So, and it's very widespread, spanning a region from Portugal to Estonia and from Turkey to Finland. The researchers showed that 38% of the variability in disease prevalence is explained by the frequency of the ACE1D gene. And a similar correlation turned up for mortality statistics also. And interestingly, the researchers also noted that the ACE1D gene is less frequent in 2 Asian countries severely hit by SARS-CoV-2. So that is also interesting, isn't it? Now, a further genetic component of susceptibility to new coronavirus may lie in the genes that encode HLA. Now, what is HLA? Human leukocyte antigens. And that is a set of proteins that keep the human immune system from attacking the body itself. Now, these proteins make up the major histocompatibility complex, or MHC, which marks self and distinguishes it from non-self. Now, in Oregon Health and Science University, Reed Thompson and his colleagues, they discovered a link between specific HLA genes and the severity of COVID-19. So, carriers of a variant called HLA-B4601 appeared to be particularly susceptible to SARS-CoV-2, as was previously shown to be the case with SARS-CoV. In contrast, the HLA-B1503 variant may provide some protection. According to the researchers, identification of a person's HLA genes, which can be done quickly and inexpensively, may help to better predict the severity of disease and even to identify those who would benefit most from vaccination. So we can now see where is the importance of this, knowing more and more about this genetic susceptibility. So several projects are underway to investigate the genetic variants that influence SARS-CoV-2 infection in greater depth. Now, there is a fantastic initiative that has been launched that is called COVID-19 Host Genetics Initiative. And this is launched by Andrea Gana of University of Helsinki. And what they aim to do is to mobilize the international community of geneticists working on this topic. Now, there is this one study is going on in Rockefeller University, which is trying to identify genetic variants that promote the development of particularly severe forms of COVID-19 in people under 50 years of age. So we know there are susceptibilities. So, elderly people with comorbidities, but surprisingly, we are also finding a lot of young people, they're having severe disease in ventilator. So this particular group of researchers, they are trying to find out why. So finding out these inequalities, like why some people are getting more affected, or whereas some people are, not having any symptoms at all, or very mild symptoms and then getting better. In United Kingdom, there were a lot of doctors who lost their life, and it was found that majority of them, a huge majority, is of BAME origin, so of minority ethnic group. So finding these reasons, these cause and effect, will help know who will need more protection, who will be, better off, all these. So findings are not cut and dried, however. Scientists who are working on these, they caution that even valid effects may be small. So with COVID-19 or SARS-CoV-2, we are still taking baby steps, very early steps. We are still starting to know a little bit at a time. And these are very uncharted territories. We did not know anything like this. So all these have to be taken with appropriate caution. So now, the knowledge about genes involved in serious disease outcomes, this can help identify therapeutic drugs. So, problem with these. So there is a problem with all these genetic studies. Now, these tend to be concentrated in populations with specific ethnic backgrounds and ancestries. So the searches for genes are most often based on biased data sets that are largely derived from people of European ancestry. So that actually omits other populations. So entirely miss those people who are at greatest risk, leaving important gaps. So one of the most significant peer-reviewed genetic studies to date, published on June 17 in the New England Journal of Medicine, they relied on 2 European populations. In that study, researchers picked through the genomes of 1,610 hospitalized patients in Italy and Spain to find DNA changes that were common among people with developed COVID-19 respiratory failure. These small DNA alterations may have only subtle effects on how a gene functions, or variants from the more typical version of the genetic sequence. So the investigators, they found many patients with severe disease had variants in a complex of genes on chromosome 3. So some of these genes encode proteins called chemokines. I know my doctor friends are familiar with this. So we are hearing about chemokines and cytokines, and we know a lot of people are dying from an event called cytokine storm. So chemokines. So these chemokines, they interact with immune system molecules. They serve as a chemical breadcrumb to attract immune cells to tissues under attack. and then those immune cells come and destroy infected cells. But with coronavirus, it's a little tweaked, and that makes all the difference. So it is found now that instead of acting for the benefit, so these chemokines actually go into overdrive and they trigger assaults that destroy the lungs. That's the cytokine storm. So scientists found a key variant that was about 1.5 times more common in people who had to be put on ventilator machines than it was among those who needed milder interventions, such as some supplemental oxygen. Severely affected patients with 2 copies of this variant were somewhat younger than those with 1 or no copy, suggesting that a double dose trumped some of the protective benefits of younger age. So we are coming back to that, why some younger people are getting severe disease when we are being told, no, it is the elderly who are more at risk. Now, the study authors say that this small DNA change is linked to decreased activity of a gene that helps regulate chemokines. The alteration is also associated with increased activity of another gene, one that encodes a protein that interacts with the molecular gatekeeper for SARS-CoV-2's entry into cells. And you know what I'm talking about. The molecular gatekeeper, ACE2, the doorknob that the SARS-CoV-2 uses to enter a cell. So this ACE2, the doorknob, is active in many tissues, including the lungs. So several of these chromosome 3 genes had already emerged as candidates of interest in early analysis. The researchers in this NEJM, New England Journal of Medicine study, also found an association with a region on another chromosome that determines blood type. This stretch of DNA sits on chromosome 9. Patients with gene for type A had 1.5 times the chances of respiratory failure compared with those who had other blood types. People with type O, though, had lower chances of such failure. So these are all coming more and more into focus. Now, we were talking about these chromosome 3. Now, the region on chromosome 3 seems to have the strongest association with disease activity. And the Andrea Gana, now I already mentioned her. She took the initiative of this global genetics search mission. So a group leader at the Institute of Molecular Medicine, Finland. So the tie to blood types is, they say, the tie to the blood types is less certain. Now, as I said, that it's very early days. So there can be some confounding factors in the studies that we are missing. We are thinking that, okay, this is related, but there may be something. So as we go on more and more, we'll be able to find out, more definitive evidence in the studies. So now about this revealing information about biological pathways. involved in disease susceptibility or resistance. These can open the way to testing drugs that can target those pathways. So there is the positivity about it. So why people without any obvious risk factor getting severe disease? So that is an extraordinarily important and interesting question, isn't it? And if we get the answer to that, we'll be very well placed in fighting this menace, this pandemic. So our understanding of this genetic susceptibility of SARS-CoV-2 infection and the severity of COVID-19 is still in its infancy. The scientific community is trying to address this issue by combining research efforts using existing genetic databases. An important step in this direction is, of course, what I mentioned in the beginning, the genetics initiative. And this initiative is intended to encourage the human genetics community to generate, share, and analyze data to elucidate the genetic determinants of COVID-19 susceptibility, severity, and outcomes. So far, this initiative shows promise because major biobanks like FinGen have expressed willingness to participate. In addition to this initiative, the UK Biobank, which contains samples from 500,000 volunteers and detailed information about their health, has now started to curate data from COVID-19 patients. In Iceland, the company Decode Genetics has monitored the spread of the virus using SARS-CoV-2 genomic analysis and has partnered with Iceland's government to sequence the genomes of viral hosts. So the patients who were previously infected with COVID-19. Now, the Greek government has recently funded an initiative, COVID-19 GR, to genotype 3,500 COVID-19 patients. Whole genome sequence, their SARS-CoV-2 genome, and performing immunogenomic analysis. All of these data will be linked with detailed clinical information collected in the Greek COVID-19 registry. Some work is going on in the United States of America. Harvard's Weiss Institute researchers and the Personal Genome Project at Harvard University, they are launching a project to compare the genomes, microbiomes, viromes, and immune systems of consenting individuals with extreme COVID-19 susceptibility and individuals that exhibit resistance. And also, there is Yale University SARS-CoV-2 Genomic Surveillance Initiative. They are sequencing the genome of SARS-CoV-2 in order to monitor the spreading of the virus in Connecticut. So, we are making progress. we need more effort, and that effort needs to be global. It is not possible to succeed with a narrow approach. And the challenge of SARS-CoV-2 is the virus is absolutely versatile. Some researchers call it evil genius, because the previous versions of this coronavirus, what they used to do was they would go into the body and immediately it's just blows over the roof. All the symptoms are there. Patient is very ill. SARS. In this SARS-CoV-2, this virus has changed in a way. The first where it launches is in the throat, and there is no symptom at all. So before someone knows there is something wrong, the virus is well inside the body and already in the process of creating that cytokine storm. So to fight something like this, which we call an evil genius, we need this all-out effort. And because of all these studies, and more studies are launching every day, we need more proper studies from the developing countries also. Big populated countries like Brazil, India, and with this collaboration, exchanging information and ideas, and involving the big corporate houses, the big pharmaceutical houses, getting the funding, we think we can win this, big, probably the biggest ever war of humankind to date. And we already know that we do not have any medicine. Nothing that is like the medicine. Maybe in the future we can have something which can work. But as we understand this virus better, the genomic sequences, and also why some of us have just the mildest of symptoms and some of us are struggling to survive. If we can analyze that subtle differences in the genetic codes and sequences, then that can be our big weapon against this pandemic. And also, not only this SARS-CoV-2, this is, to me and many of my colleagues, this actually is a warning that it can happen again. SARS-CoV-2, maybe when we find a way out, we find something with which we can deal with this, then something else comes up. But the experience of fighting this war, hand in hand, standing side by side, all of us, the humanity, would be able to fight the other ones which will be coming our way in years to come, it will be helpful. But until then, as I already said, we are doing baby steps. The only way is to break the chain of transmission. This cannot survive outside the host body for a very long time. So breaking the chain by maintaining social distancing, social isolation wherever possible, washing hands, using face masks. So all these things are very, very important until the time we find something. And as doctors, we have a huge role to play. In all countries of the world, be it United Kingdom, USA, India, Pakistan, doctors are followed, advice are listened to. So we have this responsibility to keep teaching people, educate people about the need of continuing with our precautions. Not to panic, but be careful. So that's what we should do. And Amit, my very good friend, and his colleagues are doing this wonderful initiative. I firmly believe that these talks, these Facebook Live from my esteemed colleagues, these will go a long way to create that awareness. And not only this SARS-CoV-2 or COVID-19, but following all these precautions will also help in breaking the chain of transmission of many other diseases, very age-old diseases, waterborne disease, through food, which are killer diseases in developing world. That can also help. So that can also be a very positive from this. Sorry, I deviated a little towards the end, but I thought these are very pertinent things to say. Hope you liked the talk. Thank you for being there. Thank you for listening to this. And I'd be happy to listen to any suggestions that you have. And personally will be responding if there is any query at all. Thank you. Namaskar. Goodbye. Sorry, I was also told by Amit to introduce my next speaker. So, eminent psychiatrist, Dr. Bappaditya Chaudhuri. He would be now talking about COVID-19 and immune reaction, impact on mental health. So hope you'll be there to listen to that talk. I'm also looking forward to that talk. Thank you. Bye.